Hello, everybody. Welcome back for another week of Biology number 1. So this week we are going to be doing lecture 9. It is the second last lecture of the entire course, if you can believe it. We are actually at week 12 right now. Okay, so next week will be the last week where you will be learning new things. And after that is going to be the final test. Okay, so final test is going to be on week 14. And I believe it is going to be on the Tuesday of that week. Not 100% sure, but let me just quickly double check that and see when is that going to be. You guys are 201. So the final test will be, yes, it will be on the 19th, okay, April 19th. Of course, I'll post more information as we get closer to the date. But before the final test, you also have to worry about quiz number 4, which will include lecture 8 and 9. And that's going to be happening next Tuesday, okay, for you guys, next Tuesday. That's the 12th, okay, April 12th. In any case, we have lots to do today. Lecture 9, as I said, is one of the more involved lecture. So, you know, we're going to have to be pay extra attention today. Okay. And of course, ask questions. That's very important if you don't understand something. So here we go. The gel on the right was ran after bacterial plasmid was digested using restriction enzymes. Indicate the following on the gel. All right, so a couple of things here that we should talk about. First of all, what is a plasmid? So this is a bacterium, okay, bacterium. And, you know, bacteria, they have their own DNA. Okay, so that's their core DNA, if you would, which would be kind of in the middle of the cell. And then they would have these plasmids. Okay, so plasmid are circular DNA that are found kind of scattered throughout the bacteria. And usually plasmid gives them like special features. Okay, so they might have a plasmid that allows them to grow, you know, little hairs around them so that they could cling onto each other and form a biofilm, right? There might be a plasmid that makes them resistant to antibiotics. And you know, we can actually feed them plasmids so that they would, you know, make certain proteins for us. So that's what a plasmid is. So in this question, you extract the plasmid and you're going to be digesting it. That means you cut it, right, using restriction enzymes. Okay, I think there's a question about restriction enzymes coming up later, but they are basically enzymes that would cut DNA strands at designated location. And we're going to run it on a gel, right? These are called agarose gel, which are basically polymers that forms like a mishmash of network. And by applying a charge, you're actually going to put this gel in a buffer, okay, and then that's going to hook up to a power supply, and then the current will be running through the buffers, which then cause the DNA to move down the gel. Okay, so in this picture, we have to locate the positive and the negative electrode. Okay, so the negative electrodes are going to be up here. Okay, and this is going to be the positive electrode, which means when you turn on the power supply, you're going to have a current that's going to run this way. Direction of current. Okay. Now, the backbone of DNA is made up of the sugar and the phosphate, right? So sugar phosphate backbone of DNA makes it negative, right? So that's why we want to run the current from negative to positive. It will basically repel the DNA and push them down the mishmash. Okay. If the DNA strand is really small, then they would be able to go down further because they can move through the mismatch. When I say mishmash, right, it's like this, right? These are the polymers. They basically will form all these crisscrossing stuff. And then the DNA will have to be threading through these structures. So if you imagine something really small, it's able to move through the holes easily, right? If it's really, really big, then it's going to move slower, right? So basically, the largest band is going to be the one that didn't move too far away. So that's going to be the largest band. All right, and just to refresh your memory, right? Like these are called lanes, right? So we have 4 lanes here, and we would load the DNA sample at the top here in these wells. And then once we apply the current, let it run for like half an hour or something, then the DNA will separate. The shortest band, the smallest band, is going to be the one that moves the furthest in the gel. That's going to be right there. Smallest band. Okay. The number of cuts that were made on the plasmid to produce this number of bands. All right, so let's take a plasmid. Here's a plasmid. If you cut it once, then it opens up into linear, right? So 1 cut gives you 1 band. If you cut it twice, if you go cut and cut, then you have 2 bands. 1 band here, 2 bands here. Okay, so the number of bands equals to the number of cuts for a plasmid. So you see 4 bands here. That means there are going to be 4 cuts. Right? 3 cuts for here. 1, 2, 3, 4, 5 cuts. And then 4 cuts again. Does that make any sense? So that's for plasmid. Now the question go on and ask, will the answer be different if linear DNA was used instead? Okay, and so if you have linear DNA, okay, so this is linear DNA, it's not a circle. If you cut it once, then that gives you 2 bands, right? So for linear DNA, the number of cuts plus 1 equals to number of bands. Okay, so if you cut it twice, you're gonna have 3 bands. You cut it 4 times, you're gonna have 5 bands. So if this was linear DNA and you see 4 bands, that means you had 3 cuts. Okay. Look, linear DNA, if you cut 1, 2, 3 times, then you have 4 fragments, right? Okay, so depending on whether you're loading plasmid or linear DNA, the number of bands you get will be different depending on how many cuts you make. Okay, so this process of separating DNA on a gel, anybody remember the name of this process? Help me out here. It's a really long name. Oh, there's a question. How do you know it's linear or not? I'll have to tell you, right? So here the question says plasmid, right? If it's linear, I will tell you in the question. What is the name of this this process? Anybody? Help me out here. What is it called? That's okay. This is called electrophoresis. Okay, and the purpose of electrophoresis, the use of electrophoresis is to separate DNA fragments based on size. And we can we can then determine the size of the fragments. But that's basically what it does. So again, the shorter the fragment, the further it will go down the lane. All right. Moving on. What is the reason for using tag polymerase in PCR? Just very quickly, PCR is a polymerase chain reaction. Okay. So can someone tell me what is the purpose of PCR? What do we use it for? Right, you hear PCR all the time nowadays, right? Like on the news, right? The PCR test for the COVID, right? Okay, so this is polymerase chain reaction. All right, so what is it for? Copy to copy. Yeah, exactly. Okay. It's nothing more than clone a section of DNA. Clone a section of DNA. Okay, you're gonna mass produce a desired portion of DNA. Okay, using PCR. Yes, Sakina, go ahead with your question. Yep, so in the videos, clone is same as copy. Yeah. Okay. Hold on. In the video, when cutting the specific area of DNA, you use 4 to the exponent 30. Where did you get 4 from? 4 to the 6. Wait, sorry, I'm a little bit confused. I don't. The video is from a long time ago. Could you give me a little bit of context? Is this talking about the restriction enzymes? 4 to the 6. Small. Where's small? Do we have small here? Give me a second. I'm just trying to pull up the. Can you tell me which slide it was on the test? Do you remember? Okay. I am gonna try to I'm gonna try to rephrase your question. I think this is what you're asking. Okay. Correct me if I'm wrong. Okay. So it's okay. It's okay. I think I understand your question now. So we will talk more about this in a second. Do we have a question on restriction enzymes? I think we do, right? What are restriction enzymes? Yeah, they're gonna come in a bit, but you know what? Let's just answer your question right now. Okay. Sorry, I'm rambling a little bit. I'm just trying to piece together what the question is. All right. So restriction enzymes are enzymes that will cut DNA at specific location. So for example, for a restriction enzyme, that is called small one, SMA1. It recognizes CCCGGG. Okay, so that's a palindromic sequence. It means if you read it backwards on the other strand, it is exactly the same. Okay, and then it will cut it like that. Okay, so I don't think it's 3 to the what was it? 4 to the 30. Okay, I think 4 to the 6 sounds better. So in what context did I mention 4 to the 6? I'm just saying, right? What's the chance of something like this happening randomly in your DNA sequence? Okay, so this is 6 nucleotide long, right? 6 nucleotides long, and that means I have like 6 blanks, for example. Okay, each nucleotide can be A, C, G, or T. So that means we have 4 possible choices for each blank. So if I put all the 4s here, you will see that if a strand of DNA has 6 nucleotides, there would be 4 to the 6 different combinations. Okay, if you list it all out, there will be 4 to the 6 different combinations. So 4 to the 6 is like 4,096 combinations. Okay, so what I'm saying in the video when I say 4 to the 6, I'm saying if the DNA sequences are completely random, then something like this, you would expect it to show up once every 4,096 nucleotide bases. That's what it means. Okay, so statistically speaking, right? If you have a sequence that is, let's say, I'm making this up. Let's say you have a sequence that is, 10,000 nucleotides long. Okay, and you expose it to small. Okay, small one? How many times do you expect to cut it? Well, if it's completely random, all right, this sequence you expect it to show up every 4,096 times. So if you take 10,000 divided by 4,096, right, divided by 4,096, you get 2.44. Okay, so statistically speaking, you would expect this thing to get cut twice. Okay, but it's important to understand the DNA sequence are not completely random. Okay, so even though you are probably going to cut it twice, chances are you might not cut it. You might cut it once, or you might not cut it at all. So that's where the 6 comes from. All right. So the longer the recognition site is, the less likely it is going to appear in a short DNA sequence. Does that make sense? I don't know if that was your original question, but just trying to piece together everything you said. I think that's what you were asking. Okay. Anyone else have a follow-up question on this? Okay. Let's go back here. We were talking about PCR, polymerase chain reaction, and the purpose of that is just to clone a section of DNA. Okay, so if there is a gene that you want to mass produce, you use PCR to do it. Okay, if you find a DNA sample in a crime scene, just traces of blood, very little, but you want to amplify the DNA in it, you use PCR. Okay, if you collect a nasal sample from a patient and you want to see if the person has the the antigen from COVID, right? You would amplify that specific sequence that corresponds to the COVID virus, and then that's how you know if the person is infected or not. Okay, although the kind of PCR they use is different. It's called RT-PCR. So you actually have to start with RNA and then you work your way backwards. But in general, PCR is amplifying, cloning, copying a section of DNA. So what is the reason for using tag polymerase? Okay, so tag polymerase is heat resistant. Okay, you actually, the tag polymerase was discovered from bacteria that live in hot spring. Okay, so their polymerase can withstand hundreds of degrees without breaking down. Right. So why do we need this? Well, because as part of the PCR procedure, okay, so PCR steps requires DNA sample to be heated up to high temperatures. Okay, around 70 degrees to 90 degrees, depending on your sequence, depending on the sequence that you're trying to amplify. And so if you use normal polymerase that we have in the body, they would totally break down at that temperature range. Okay, so that's why we need the tag polymerase. All right, so what are the 3 major steps in PCR? Okay, so step 1 is denature. Okay, you have to separate the DNA strand. All right, so here is your original DNA. Right? So step one, you would crank up the temperature. Okay, increase temperature. By increasing the temperature, you're breaking the hydrogen bond and the two DNA strands will separate. Okay, so that's step number one. And that's the step that you have to heat it up to like 70 degrees or 90 degrees, right? If it has more C and G, you need to heat it up higher, right? If it's, mostly A's and T's, then you don't have to heat it up as much, right? Because G and C has 3 hydrogen bonds, right? We talked about that before. So you're gonna heat it up, okay? And then step number 2, it's called anneal. Okay, so this is where you will lower the temperature a little bit. Okay, not so much that the whole strand is going to come back together. You're just going to lower it a little bit, which allows the primers to bind to the template DNA. Okay, so step number 2, you lower the temperature a little bit, and that will allow your primers to bind to the strand. Okay, so you design the primers, right? Like on the computer, you would design the primers, so you would know exactly where it binds to. Right, so like that. So now I'll be cloning this section. Okay, and finally, the last step, step number 3, is going to be the elongation. Number 3, elongation. So you lower the temperature even further, and that would allow the copying of DNA. Okay. So there is the original DNA. Here are the primers. And what happens is you will clone the DNA. So after one cycle, you would have 2 copies. Right? 1 copy, 2 copy. And then you will repeat the process. You would heat it up again. And then these would separate. And then afterwards, you would end up having 4 copies. And then afterwards, you would have 8 copies, 16 copies, 32 copies. So after just 10 cycles, you would have thousands of copies. Okay. So like, 2 to the 10 is 1024, right? So after 10 cycles, you would have 1024 copies. But usually we don't do it for like 10 cycles. We do it for like, 20 cycles or whatever. So like by the end, you're going to have like a bunch of the DNA. Is that okay? Any questions about PCR? Okay, so here's a question about restriction enzymes. What are they? How do they work? Restriction enzymes are naturally occurring enzymes found in some bacteria. Okay, they are basically defense mechanism. A defense mechanism to shred foreign DNA. Okay, so if foreign DNA gets into the bacteria, they don't want it there. Okay, they're just gonna cut it up. Okay. It does not cut their own DNA because their own DNA are methylated. Okay, so like if you look at the DNA of the bacteria, they are gonna have like CH3 on them. These are called methylation. So these so these methylation CH3 protects the bacteria own DNA from restriction enzymes. Okay. Now, how do they work? We sort of mentioned this earlier. Restriction enzymes recognizes specific DNA sequences. Okay. Usually palindromic sequence. Okay. So a palindromic sequence is a sequence that reads the same forwards and backwards on opposite strands. So let's say we have ATC, GAT. Okay. On the other strand, it would be ATC, GAT. Right? So if you read it this way, it's same as reading it backwards. So that's a palindrome. English is same thing, right? Like ADA, right? That's a palindrome. Forward, backwards, it's the same, right? That's the only one I can think of on top of my head. But yeah, like wow, wow is another one. Very good. Okay, but there are like longer ones, right? Okay, those, yeah, level. Thank you. Oh, you guys are expert in this. That's right. You guys, these are palindromes in English, and this is palindrome in DNA. All right. Now, once the restriction enzymes identify a palindrome, it could cut it one of two ways, depending on the restriction enzymes that we're talking about. Alex says race car. Race car. Yeah. Race car is another one. Yeah, we should have a palindrome contest. So sometimes you will cut it like this. This is going to create what we call blunt end. Blunt end. Okay, not very useful for biotech. And then you could cut it like this, right? So that is called sticky ends. Sticky ends. Okay, so sticky ends are more useful, as you might recall from the lecture recordings. Any questions about restriction enzymes? No? All right. Next, we have short tandem repeats, or STRs. What are short tandem repeats? Tandem, if something is in tandem, it means it's right after the other, right? So these are repeats that are right after each other, and it's short repeats. Okay? So for example, you can have a STR that's like AT5. So AT5 means it's AT, 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 AT. AT repeated 5 times. Okay, you can have like a AT10. AT10, which is like AT, 10 times, right? So I was gonna be lazy and copy this, but I think this is gonna end up wasting more time than if I write it out. But I'm committed, so I have to see it through. So there, it's 1, and then there we go. So 10 times. Okay, so that's a short tandem repeats. Okay. The thing about tandem repeats is like if it's a lot of repeats, it's easy for the polymerase to make a mistake when you copy it. Okay, and so there are 10 tandem repeats here, but then when it copies it, they might by mistake add an extra one in it. Okay, so it becomes like AT11, or they might miss one and become like AT9 or something. Okay, so from one generation to the next generation, the tandem repeats can actually change. Okay, but people who are related tends to have tandem repeats that are in the same location. Okay, so I think in the lecture I talked about CAG, right? Okay, so CAG is a tandem repeats that is associated with anybody remember what condition I was talking about in the lecture? Did I even talk about this? I usually talk about it. I don't remember specifically if I mentioned it, but anybody help me out here? CAG, what kind of disease it's related to? Sita, do you mean like HTT? Yeah, Huntington's, okay. That's right, Huntington's disease. Okay, so this disease, it's a neurological disorder that does not show up until after, I think like age 40 or something like that. Okay, so by the time people show signs of Huntington, they would already have kids already, and so they would already pass on their genes, and CAG, if the repeat is bigger than 35, then there is a high chance that the person is going to have the Huntington disease. Okay, so anything below, it's going to be okay. So people have CAGs, let's say in the 30s or something, or 31s or 32s. They might not have Huntington's, but as I said, right, the mistake could happen when you copy the DNA. So it might show up a few generations later. So there you have it. That is the short tandem repeats. Next, we have RFLP, right? All these acronyms. What is normal? Normal is anything under 35, right? For the CAG. Okay, for the CAG, anything under 35 is normal. Okay. What is RFLP? RFLP stands for restriction. Restriction fragment length polymorphism. Now that's a mouthful. Let's try to deconstruct this thing. So polymorphism means it's just fancy word for saying different. Okay. So a polymorphism between you and I just means that our DNA looks different at a specific location. Okay. And what kind of difference are we talking about here? We are talking about the difference in the length of the fragments created by restriction enzymes. Okay, so that's what it means. Now let's do an actual example. I'm sure I talked about this in class in the recordings, but you know what? It's complicated. So let's go through it again. Okay, so let's say that there is a restriction enzyme. Okay, I'm totally making this restriction enzyme up, and this restriction enzyme is going to recognize the sequence TTAA. And so that's a palindromic sequence, right? Because if you go backwards, it's like that, right? So let's say this person on a particular chromosome, doesn't matter, chromosome 4 or something. On chromosome 4, there is going to be the TTAA here. Okay, so on the other strand is going to be AATT, right? Okay, so this is person number 1. Person number 1. All right. And so if I treat it with a restriction enzyme, this particular restriction enzyme, it is going to cut it. It doesn't really matter how it cuts it. We'll just say it cuts it and create a blunt end. There would be 2 fragments. Okay. And so if I run the sample on a gel, right? If I do electrophoresis, run the sample, I would expect to have 2 fragments, 1 fragment, 2 fragments. Okay. Any questions so far about what's going on here? Please ask before I talk about person number 2. Everything makes sense? All right. So for person number 2, this guy, or girl, we don't know, this person is going to have a different nucleotide. Okay, so instead of TTAA, maybe this person has TCAA, and on the other side it will be AGTT, right? And this difference could be as a result of, you know, like a silent mutation or something. It could be a bunch of other reasons, right? We talk about these SNPs before, remember? The single nucleotide polymorphism, right? A difference at a single location. So if this person has a different nucleotide here, and you treat it with this restriction enzymes, there will be no cuts because this sequence does not occur on this person. And so it's just going to be one fragment. Okay, and if you load it on a gel, if you load it on a gel, then you would just have one big fragment, one sequence. Okay, so just by looking at this, I'll be able to tell, you know, whose sample is who, right? So let's say I find a blood sample at a crime scene. Okay, so I extract the DNA, I do PCR to clone this section. Okay, you have to know where these are located, right? And you know the human genome map is located online, so you can find these tandem repeats easily, find these locations easily, and then you're gonna expose the DNA, the clone section to the restriction enzyme, and then you can load it on a third lane. Okay, you collect sample from each person, and then you're gonna run the DNA sample, and then you see that, oh, there are two bands. Then you will say, oh, the blood belongs to person number 1 and not person number 2. Okay. That, of course, is an oversimplification of what really happens, but you know, in a nutshell, that's basically what it can be used for. Is that okay? Any questions? All right, no questions is good. Next, describe how a gene can be inserted into a plasmid. Okay, so we're gonna assume the gene has already been cloned using PCR. Okay. So let's say this is my gene. And that's going to be my genes. And then the genes are going to be flanked by some palindromic sequence. Let's say, again, AATT. It's just an easy example. Okay. Now, in order for you to be able to insert the gene into the plasmid, you need to buy a plasmid, and you can buy them online, okay? Like there are a lot of biotech companies out there, Fisher's, Sigma, all these places, you can buy plasmid, okay? And in order for you to insert the gene into the plasmid, you have to make sure that the plasmid contains the same recognition site as your ends. Okay, so these recognition site. Okay, should be the same. Okay, so what happens is we are going to treat the plasmid and the gene with the same restriction enzyme. Okay, the one that I created, right? That will recognize the AATT. And so you need to choose a restriction enzyme that will create sticky ends. Okay, so we're gonna cut it like this over here, and then over here it's gonna get cut like that. Okay, and same thing over here, right? We're gonna cut like that. Okay. Sorry, I didn't do a good job there. Let me try that again. Like this. Okay. And then like this. Yeah, the A here, right? There we go. Okay. And then same thing over here. Right. No one's gonna ask you to draw these things, right? Like I'm just trying to explain to you, right? So don't feel intimidated because you're like, oh, I cannot, I don't know how to draw these exactly. Now, what's gonna happen is this. Watch. All right, we're gonna cut this open, and now we're gonna have the plasmid. Okay, and on one side we will have like A and then TTA. Right, basically this half. I'm gonna try to highlight it to show you. And then on the other side, let's choose a different color, which is going to be ATT and then A, like that. Does that make sense? You just pop it open, right? Because of the cut that was created by the restriction enzyme. Now, what about this guy? What about this? Well, we will have our gene of interest, and then the end will look like this now. Okay, this part, it's gone. So I will have the A, and then the bottom would be TTA. And on the other side, this part would be gone, and I would have like ATTA. Okay. And then you can see, this is the exciting part. This part here will be able to bind here. And then this part here will be able to go right here because they match, right? Okay, and that's why you need them to have the same recognition sites. Otherwise, it will not be able to go in. And so afterwards, right, we will add a little bit of ligase, right, to seal the deal. And we would have the gene inserted into the plasmid. Does that make sense? Are we together? I mean, it's very complicated. I try my best. It's much easier to explain this on the board than to do it, remotely on a computer screen. But I hope it makes sense. Any questions? Don't be shy. All right. Now I know what you guys are scared of or worry about. Like on the test, right? You'll be like, well, how are you gonna ask questions related to these things, right? Like what are like how are we supposed to study for this, right? Okay. Well, just kind of know the general steps, right? Like if I ask you to organize the steps, right, into like if I give you a bunch of steps of how to clone the DNA, right? You should know what the steps are. So for example, right? Let me see here. Let's say I give you something like this. Okay, so here are the different steps. All right, can you guys see? Is it too small? Let me make it a little bit bigger. Oops. There we go. Okay, so if I ask you to organize them into the right order, what would it be? Okay, I'll give you a moment to read through all that, and then you can tell me. Okay. What do you call for these steps? What do you mean? What do you mean? What do you call for this step, Christina? No. Steps for, oh, this is for like cloning. Yeah, cloning, right? Cloning a gene, feeding it into a plasmid, right? Okay, it's the same thing we just talked about. Okay, so these are all the steps. Okay, so what's the right order? Okay, anybody? Any takers here? Want to try? Tell me what the right order is. What is the first step here? What do you think? Yes, number 3, right? Extract plasmid DNA from bacterial cell. We gotta have a plasmid, right? But you know, nowadays we don't really do that. Nowadays you just order it, you just buy them. Okay. They're like what, like $40 or something, a plasmid. Okay, so you can just buy them online. But before that, you have to, and I mean like the people who sell them, right? They probably have to extract it from the bacteria or something. Okay, actually not really. They can actually build it from scratch using nucleotide, but whatever, it doesn't really matter. Okay, so in this question, you can extract it. So that's step number 1. Okay, 3 goes first. And then what? What are you gonna do? After you extracted the plasmid, what should we do? Look at the chicken scratch diagram that I've created here, right? Okay, Cedar said cut the plasmid DNA using restriction enzyme. That is absolutely correct. You gotta pop it open. Okay. Then what? After that, what do we do? Okay. After that, you're gonna have to do number 4. Hydrogen bond the plasmid DNA to the non-plasmid DNA fragment, right? So you gotta put this in there, right? So I mean, this question actually is missing the part where you have to treat the clone DNA with the restriction enzyme as well. But given the constraint of the question, that's what it is. And then you're gonna use a ligase to seal them, right? And then finally, you're gonna feed this plasmid back into the bacteria. All right. So if you see something like this on the test, you will see something like this on the test. Okay. Hint, hint, nudge, nudge. This is the answer. Okay. 3, 2, 4, 5, 1. Are we good? Okay. I assume the rest of you are also okay. Thank you for verifying, Adriana and Sakina. Okay, great. Thank you. Thank you. Appreciate the feedback. By the way, talking about feedback, right? There should be like a course evaluation that you can do for me, right? You probably have received an email already. So please take the time to review, to complete the survey. Is anonymous. I would not know who says what, so please be honest with your review. Obviously, it's very challenging moving the whole course online. It's challenging for you as well to learn from online, right? So, if there is something that I'm not doing too well, please let me know how I can improve. Yeah, it's completely optional, okay? But I would really appreciate it if you take the time to do it. It probably would just take you 5-10 minutes. There is no prize for doing it. There's no draw for like $100 Amazon gift card. But your input is very important, right? It will help me improve. And you probably will see me next semester, right? Like unless you deliberately choose not to be in my class. But I will be teaching some of the second semester. So, some of the review is probably going to help me do better. Okay. I actually have been teaching at Centennial for 12 years now. So sometimes I get tunnel vision, right? I get, it's just too easy, sort of, right? Because I just do the same thing over and over again. So if you have suggestions of how I can improve, how I can do better, that would be wonderful. Again, it's anonymous, so I wouldn't know it's you if you make a comment. Okay, so if you say something bad, that's okay, right? As long as it's not like rude, right? you can tell me how to improve. Constructive criticism. Okay. Do we have the choice to pick professors for next semester? Yeah, I think so. I mean, like I don't know, right? But usually you can see who who teaches the course, right? Okay, so like in a way, maybe you can choose that. Okay, so yeah, maybe you can avoid me if you think my tests are too hard, or maybe you want to come back if you like my corny jokes. Okay, whatever it is. But for the summer, I will only be teaching the first 6 weeks, and then I'll go on summer vacation. Then you will get a different prof anyways. Yeah, anyways, next question. Describe how to determine if the bacteria have accepted the plasma you transform into them. Okay, so transforming a plasmid means you feed it to them. Okay, but how do you know? How do you know they have taken up the plasmid or not? Right, like you cannot really physically see them. So to answer this question, we have to look at the plasmid itself. So usually the plasmid, okay, so you're gonna have the gene here, and that you have just inserted. And on the plasmid, there are usually another gene that is going to give it some kind of resistance to an antibiotic. So over here, they might have like a amoxicillin resistant gene. Okay, so like AMOX resistant gene. All right. So if you feed this to a bacteria, okay, if you feed this to a bacteria and it takes it up, not only will it start making your gene, it will also start making the amoxicillin resistant protein. Okay. And so after you transform the plasma to the bacteria, and you can transform the plasma to the bacteria using various methods. One way is just to give them a little bit of electric current, okay, that causes their membrane to become a little bit loose. And if they don't die along the way, then the plasma might go in. Okay, it sounds very cruel, but it's okay, it's bacteria. So what happens is after you do the transformation process, some might take it up, some might not, okay, and some might die. And then you will grow them, okay, you will grow them. So for those of you who don't know, you can grow bacteria on these agar plate, okay? So these are called culture, okay? And it's agar in there, and you would give them a little bit of broth, okay? So the industry standard is called LB broth. Okay, so it's a nutritious broth that the bacteria love, and they're gonna grow on it. So if you just grow them on a regular culture, then you will see them, grow into colonies. Okay, you actually would spread a few drops on them, and then you put them in an incubator that's 37 degrees, and then you go home, and then you come back the next day. You will see the colonies. Okay, so this is going to be a culture with just LB broth. Okay, so no antibiotics. Now, if you have another culture here, okay, everything is the same, but this time is LB broth plus the amoxicillin. Okay, and then you put the bacteria on them, and you see, there are some growth. All right, only the ones that have received the plasmid would be able to grow, because all the other bacteria that did not take up the plasmid, they would not have the amoxicillin resistant gene, and therefore they would not survive in the presence of amoxicillin, right? So the fact that you see that they're growing here, all these has to come from bacteria that has been successfully transformed. Does that make sense? Okay. And so you can scoop them up with a little loop, right? This is true because I've done it. Okay, like you scoop them up with a little loop, and then you can just mass produce these guys. Okay, so that is basically how you select for them. All right. Moving on. If you don't have any question. Explain how STR and RFLP can be used to compare DNA sample. Well, earlier I already talked about how to use RFLP to compare DNA sample, right? So maybe I'll do STR. All right. So let's say we have a individual number 1 here. Okay. You know what? Let's call this Bob. Okay. So again, the entire human genome is available online. There's a database. So we can easily identify the location of, STRs of interest. And just to refresh your memory, short tandem repeats are these nucleotides that repeats themselves, right? Like AT, 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 or like CCC, GGG, CCC, GGG, or something like that, okay? So let's say on chromosome 2, there is going to be a repeat of A and T. And then for Bob, he's going to repeat 10 times, okay? And how do you know he repeat 10 times? Usually, the tandem repeats, we could find the sequence that's flanking it, okay? And then we can just run PCR to clone that specific section. And depending on how long it is, we would know, how many times it's being repeated, okay? And so on a separate chromosome, let's say on a separate chromosome, this is chromosome 14 or something, and then at this location, okay, there is going to be a repeat for like ACG or something, repeated 5 times this time. And then there's going to be a third chromosome, whatever, chromosome 20. And then there's going to be, CCG here repeated, let's say 20 times, okay? So Bob has these short tandem repeats in his genome. All right. So what we can do is we will design 3 sets of primers. Okay. So one set that flank the AT. When I say flank, it means like they are on the side of the gene A and T, right? Okay. It's like this. As an analogy. Let's say I give a textbook to you, and I say, please go to the library and photocopies, from here to here, okay? And instead of telling you what the pages are, I just put the bookmarks. I put 2 bookmarks, okay? So the bookmarks are kind of like the primers. And the bookmarks would tell you to basically photocopy everything in between these bookmarks, right? So in a way, when you design a primer, your PCR is going to clone everything that's between it, okay? So we're going to clone all these A's and T's, right? And we're going to design primers to clone everything between here and here. So I have the ACG repeats, and finally we're going to have a third set of primer that would do that. Okay, and so that's going to be person number 1. And let's say we copy all of those tandem repeats, and then we're going to run it on a DNA gel, right? Okay, on an agarose gel. What you will see, for example, is, okay, let's say this is the first one, and it's repeated 10 times. So I would expect it to see a band like, this is 10 times, and there are 2 nucleotides, so there is a total of 20 nucleotides. Over here, that's 5 times 3, so that's 15 nucleotides. And here, the last pair here is 20 times 3, which is 16 nucleotides. So I might expect a really really long one that corresponds to the 60 at the top. I would expect like the red one to be, the furthest down, and then you would expect a pink one that's really close to it. And when I say red and pink, they're not going to be colored, right? I'm just trying to color code it to show you how it would correspond to these tandems, repeats. Does that make sense? Any questions about what I've done here? Okay, so let's say we have another person now.
Just one question. Sorry, why this is more repeat but short?
Why is what? I'm sorry. Could you repeat the question?
20 times repeat CCG but short.
But short?
Yes.
Why is it short when it's 60?
Yes.
Well, compared to the whole DNA, right? Whole DNA is like, thousands and thousands, right? So, 60 is actually quite short. But the point...
Compared to these 3, like first, second, third, there are...
Compared to these ones?
Yes.
Yes, compared to these ones, this is longer, right? That's why this one is going to be on top of the gel, right? It didn't go very far, right?
But when you draw the picture, it looks short. That's why I just...
When I draw the picture, it looks short. You mean like this picture?
No. Left side.
This picture?
3 lines, yes.
The 3 lines? Oh, no, 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 no. This is the whole DNA, the whole chromosome 20, right? So like, like this. This is the chromosome, right? Okay, this is the chromosome. So the repeats is just on part of the chromosome, right? Do you understand?
Okay, got it. Thank
Yeah,
you.
yeah, yeah. Like chromosome 20 is shorter than chromosome 14, right? That's what it is, right? Okay. Okay, so just one more time, right? Okay, just for those of you who are a little bit confused here, I know this is confusing. These repeats can occur anywhere in the DNA, okay, anywhere. So we have chromosome 1 all the way to chromosome 22 plus the sex chromosome. So on these things, we can have, tandem repeats anywhere. For the sake of simplicity, we are just looking at 3 locations. I'm saying at chromosome 2, maybe there is a lot of repeats here at, at this location, and it's AT repeated 10 times, okay? So to clone this, I would put a primer here and a primer here. So after PCR, I would have a bunch of ATs that are repeated 10 times, right? Okay, so 10 times 2 means that the fragment is 20 nucleotides long. So when I run it on the gel, the 20 nucleotides will show up like over here. The 15 here, right? 3 nucleotides repeated 5 times is going to go a little bit further, but not that much further because 15 and 20s are pretty close together, right? Okay, but then the 60 will be all the way up here. Okay, so that's basically what this is talking about. Now, for another person, okay, let's say we have like Angela here. Okay, so Angela, we're going to look at the same location. Okay, so on chromosome 2, number 2. Okay, I'm not going to draw the chromosome this time. Maybe her tandem repeats is AT 15. Okay, on chromosome 14, maybe it's ACG 10. And then on chromosome number 20, maybe CCG, I don't know, like another like 7 or something. Okay. So what happens? Again, we will use the pink primers for this one. We will use the red primer for this one, and then we will use the purple primer for this one. Okay. So if we do the math, we will see over here, 15 times 2 gives me 30. 10 times ACG gives me 30 as well. Okay. And then 7 times 3 gives me 21. 21. Okay. So if I run her sample on a different lane, right, we would see, 21 to be like further down. So maybe around here, right, corresponding to the 20 location. And then the 30 and the 30 here, they probably would be around the same place. Okay, they would be at the same place. So you would have sorry, this one was the purple one. You would have a pink one here, and then you would have a red one here. Okay, so that's how you can tell the difference, right? If I have a third sample, I can compare it to this person versus this person, and that's how I can tell. Now, in real life, we are not just going to do it for 3 separate chromosomes. Okay, in real life, we are actually going to do it for multiple locations. So what happens is, rather than having like 3 bands or 2 bands, you're actually going to have like a lot of different bands. Okay, and that creates like a barcode that's unique for a person. So on for a different person, they might have a different set of patterns. Okay, so that's why it's very unique, right? And it is as unique as your fingerprint. So that's why this is called DNA fingerprinting. Okay, does that make sense? Okay. You know what? I think we should take a break. Okay, just give your brain a bit of a rest, and then we'll continue afterwards. Okay, so see you in 5 minutes. Hello, it's me again. Let's keep going. Hey, next question. What are some challenges related to the use of gene therapy? So gene therapy basically involves, for example, replacing a defective copy of a gene, right? And you know, some of the challenges that is related to gene therapy would be target delivery. What that means is, how do you make sure that the gene that you're trying to insert is actually inserted at the intended location? Okay, how do we make sure that you are able to replace the defective copy at the exact location that you want it to be? Okay, so that's one of the challenges of gene therapy. We actually made a lot of progress in this area in the recent years because of something called the CRISPR technology. So CRISPR allows you to have precise control of how you edit the DNA sequence. But that's, you know, for another day. We also have concerns with immune response. Okay, sometimes the gene therapy is the gene that you're trying to insert is delivered using a virus, and you know, for some people that could, you know, trigger some kind of immune response and rejection. That could be an issue. We also don't know how the gene you've inserted might cause off-site interaction, and so interactions with other genes. Okay, and finally, cost. Again, I don't know if I talk about this in the lecture. I don't remember. I get confused because I have another course that also talks about this stuff, and you know, I don't remember which one I use what example. But there is this famous kid, well, not really a kid, but like a teenage person. He was blind. And he actually went on American Idol, I think, and he, you know, had this beautiful voice, and you know, he won the contest or something like that, but he was blind. Okay, and he actually undergone gene therapy, and he gained his vision back. Okay, but the cost for something like that goes up to millions of dollars, 1.4 millions, I believe, US dollars to do that. So not everybody can afford something like that. Obviously, he was, you know, probably fortunate because he's like a well-known person. So maybe, you know, they get sponsorship for doing that. But most insurance companies not going to pay for something like that, right? So an average person might not be able to afford something like that. And you know, gene therapy is going to be very personalized type of treatment. So you know, we don't have a good way of funding this yet. Sita asks, cell therapy, is it similar to gene therapy? I'm not sure what cell therapy is, but the gene therapy might involve modifying a cell and then putting that cell back into the body. So for example, right, and this is something that is being done. Researchers can take your white cells out of the body. They could, you know, do gene editing on the white cells so that they would be able to identify cancerous cells, and then they would grow them in the dish and then re-inject them back into the body, and they would go seek out cancer cell and destroy them. Okay. Alex asked, what was the durability of his sight after the surgery though? Yeah, I think he had a complete recovery. Okay, I mean, I haven't looked up the guy on Google recently, but as far as I remember, I think it was a long lasting. But you bring out an excellent point, Alex. The treatment, like you know, some companies say you don't have to pay unless it works, right? But what's considered as worked, right? Like what if it worked for a year and then it stopped working? Am I going to have to shell out another $1 million for another therapy, right? So that's obviously another concern. But you know, if you Google American Idol blind contestant gene therapy, you probably will find a guy. I totally don't remember what his name is though. Christina asks, stem cell used for treating diseases? Yeah, but that's a different story. Stem cells are these cells that can become any kind of cells in the body. And so they could be used for treating things like leukemia, which is a little bit different than what we're talking about here. If any of you is a mother, then you might remember when you gave birth, the hospital might give you a form and ask you whether or not you want to save the cord blood for your baby, right? Okay, they can extract a little bit of a cord blood from the umbilical cord, and that contains a lot of embryonic stem cells for your baby. And the idea is, if you save those stem cells one day, if your child becomes sick for certain groups of disease, those stem cells might be able to cure them, right? I was offered that option, but it's like a couple $1,000 initial upfront fee, and then you have to pay every month to keep them frozen or something. And yeah, it was too expensive. I mean, I don't have my own stem cells, so we didn't do it for our kids. Anyway, maybe you know what I'm talking about, maybe you don't. But that's an option. Alex said, at work, I overheard a couple that took their placenta home to consume. You know what? I actually have heard of stories like that. I'm not sure about the human ones. Like the hospital might not let you do it. Like if you give birth at home, then maybe you would have access to your own placenta. But I've heard of people extracting things from like animal placenta and use it for like de-aging or rejuvenating purposes. It's a quite big of an industry. Talking about placentas, just a funny story, just to keep you guys less bored. I'll be quick about this one. So I read on the news, I think it was last year, that they found a placenta in some Toronto park, and the police were freaking out because they were like, oh, maybe someone gave birth and abandoned the baby, so they were trying to find the baby and all that, because they saw the placenta, right? But no babies were inside, and then they search and search and search, and then they made it onto the news. And then the woman who threw the placenta away, she saw the news and she was like, "Oh my God, I was the one who threw away the placenta." So he contacted the police, and the story was she gave birth like decades ago, and then she didn't know what to do with her placenta, so she put it in the freezer. And then now many years later, she finally decided to throw it out. And then she was like, "Oh, it would be a waste to throw out this, perfect piece of placenta. Maybe I'll just leave it in the park for the coyotes to eat it." And that's what she did, right? But the police who got the call about the placenta, they thought it was someone giving birth and abandoning the baby. Anyway, moral of the story: if you have a placenta in the freezer, please do not leave it in the park. And you can search up that story. It's a real story. It was that well, if you freeze it, right, like it's gonna get preserved, right? I mean, I sometimes kind of gross when I dig up the bottom of my freezer. I would find stuff that was like a year old, and I always feel like I shouldn't eat it, but it turns out to be fine. So yeah, anything you freeze at like -20 or whatever turns out going to be okay. Oh yeah, you burn the placenta. Is this some kind of ritual, like ceremonial purposes that you burned it? Wait, did you say burn or burn? Oh, bury. Sorry, I read that wrong. Okay, buried. Okay. Yeah. All right. Anyways, moving on. We have the next question. PCR requires two. No, I just read it wrong. I thought you said burned, but buried. Yeah, that makes sense. PCR requires two DNA primers to be used. One that is complementary to 5' and then the other would be complementary to the 5' on the opposite strand. Predict what would happen if only one primer were used. Okay, so if you only use one primer, then the cloning process will be linear. rather than exponential. Okay, because you're only copying it once every time, right? Okay, so let's say 10 cycles. Okay, if you do this, you would only have like 10 copies. Okay, compared to 2 to the 10 copies if you do it properly. All right. Describe how electrophoresis worked. I already talked about it. I'm not going to go through it again. Give 3 examples of how gene therapy can be used to treat disease. Number 1, it can replace a non-functioning gene, non-functional gene. You know, for example, right, like sickle cell anemia is a prime target for using gene therapy. The sickle cell gene has one single nucleotide mutation. So if we're able to deliver a normal version of that gene, then you're going to be able to, you know, essentially cure sickle cell anemia, right? Number 2, we can suppress a gene. Right? If a gene is overly active, you know, promoting growth of a cancer cell, for example, then suppressing it would be beneficial. Now, you should understand that the gene therapy is not likely going to be able to reverse some of the damage that's already done, but it can stop the progression of the disease, right? Number 3, we can also modify the clinical outcome, right? And that's what I meant when I say you cannot reverse the disease, but you can alter how the disease is going to progress. All right. All right, and now the part that everybody is worried about, application questions. You should try the practice quiz for the biotech section. Okay, if you haven't been doing them, you should definitely do it for this one, because it's just, you know, complicated stuff, right? So make sure you try those. It will give you an idea of what kind of question to expect on the test. So in this question, it says the recognition site for BAMH1 and BCL1 is shown below. Okay, so these are restriction enzymes, and these are the sites that they are going to be recognizing. So we have the following DNA being treated with BAMH. How many DNA fragments would you expect? All you have to do is look for GGA TCC on the sequence. Okay, and you can see that, you know, just scan through the sequence and then you will find, oh, GGA TCC right here, right? And I think that's the only one. And so if you treat this with BAMH1, it's going to cut it like this, and we will have 2 fragments. Okay, 2 fragments. Okay. And then the next one, if you treat the DNA fragment produced in part A with BCL1, what results are you expecting? Okay, so this thing was there before. So that's already cut. And now I'm going to look for TGA, TCA. So TGA, TCA is right here. Okay, and so this would be cut again. Right? No recognition site here. So if you treat it with both restriction enzymes, you will end up having 3 fragments. All right. That's it. So here it says, suppose you run the DNA sample containing the above DNA fragments on an agarose gel. Draw the expected result on a gel. Okay, so here is my gel. It's really fun to make the gel. Okay, You take the powder out of a bottle, add some water, some buffers, put it in the microwave, do it for about 10 seconds, and then you pour it into a mold and you get the gel. So here is lane 1, 2, 3, and 4. Okay, so untreated, just the DNA sample alone. So just the DNA sample alone is just one long strand, and you probably will find it near the top. Okay, DNA sample treated with BAMH1. So BAMH1, as we saw, is going to cut it into 2 parts. You will get a short one and then a relatively longer one. So you would have one fragment at the bottom and then another fragment here. Okay, so that corresponds to the longer one, and then that's the shorter one. If you treat it with just BCL1, so only this get cuts, right? So you would have a longer one and a shorter one. Okay, so probably similar to number 2. And if you treat it with both, then you would have 2 short fragments and one, relatively longer one maybe. So 3 fragments like that. Any questions about how I come up with this diagram? Can you repeat it one more time? Sure. All of them or a specific one? Okay, so here, right? All of them. Okay, sure. Let's just take the original fragment again. Okay. So this is the original, right? Original. If I don't treat it with anything, if I just load this one, you will only see one band. Does that make sense? Okay. If you don't add any restriction enzymes, then there will be no cut, and therefore you only have a single band. Does that make sense? Okay, so that's why in lane number 1, there's only one band, right? Lane number 2, we will treat it with BAMH1. So BAMH1, right, it's the one that I use yellow to represent, right? So if you treat it with BAMH, it will find the GGA-TCC, and then it will make a cut. So you will have one fragment here. That's one fragment. And then you will have the other fragment here, right? So one long one and one short one. So if you treat it with BAMH1 and then you load it, you would have 2 bands. This band corresponds to the long one, and this band corresponds to the short one. Let me make it smaller here for you, right? So the long one here, that's the top one, and then the short one, bottom. Is that okay? Now, number 3, it says if you treat it with BCL1, right? Okay, so BCL1, so again, this is original, and if I treat it with BCL1, then only this part gets cut, right? Okay, so now I would have 1 fragment and 2 fragments, longer and shorter, so longer and shorter again. Okay, but if you do it twice, let me just put it back in here so it will make sense in the solution when I post it online. Now, sorry, give me a second. Now, if you do it twice, if you treat it with both of the restriction enzyme, then it will cut it once here at the BAMH site, and then it will cut it again at the BCL site. So you will have a total of 1, 2, 3 fragments. Okay, and that's why there's 3 bands here. Okay, hope that makes sense. No problem. Okay, next part. A linear DNA sequence, right? So, Sita, you asked me earlier, how do you know if it's plasmid or linear, right? So it will tell you in the question. Okay, a linear DNA sequence is treated with 3 different restriction enzymes in 3 separate vials. okay? And then you load them into 3 different lanes, okay? Based on the results, how many times did each restriction enzyme cut? Okay, so over here you see 1 band. So how many times did it get cut? Anybody? It's a linear DNA and you see 1 band. How many cuts? No cuts, that's right. No cuts. If you see 2 bands, that's 1 cut, right? It's not hard to visualize, right? There's 1 band, you cut it once, you have 2 bands, right? Take a rubber band and try it out yourself, okay? And then 3 cuts, 3 bands, 2 cuts. Someone tell me what is the purpose of the markers here? Why do we have the markers? BP is a base pair. Yep. So what do we use the markers for? Okay. So you can actually buy the markers, again, of all places, online. Okay, and the markers comes with a bunch of DNA fragments of known size. And so when you run the marker in parallel to your lanes, right, you will be able to see. Okay, for example, you get a band here, right? You just compare it to the marker, right? The marker says, oh, this one is 1,000 base pair. So this one must be 1,000 base pair as well. Okay, this one here, right, correspond to the 800. Right? So the purpose of the marker is to help you determine the size of your fragments. If you just run the lane, run the DNA without the markers, like I don't know how long that is. I don't know how long that is. I don't have a reference, right? That's why the markers is there to give you a reference. Okay. All right. This is also a very confusing question. Let's see what's going on here. You want to insert a gene in the following plasmid. So there's the plasmid. Let's take a look at what these are saying. Okay, so these things on the outside, those are the restriction enzyme site for the corresponding enzyme. So PST1 will cut here, EcoR1 will cut here, and so on and so forth. The ORI, that's just the origin of replication. Okay? It means if you insert a gene here, it's gonna get cloned this way. Okay, so that's where transcription is going to start. Okay, and then these sites, those are the antibiotic resistant gene, right? So you have the ampicillin resistant gene here, you have the tetracycline resistant gene here. Okay, and that's just the name of the plasmid. All right, so if you want to buy them online, you'll be like, oh, I want to order the PBR322. All right. First, explain how you will amplify the gene you are trying to insert. Okay, PCR. We talked about that already. According to the plasmid map, what do these things represent? Okay, why is it important to know the location of these sites? Okay, so these are restriction enzyme recognition site, right? It's important to know the location because that will tell you where your genes will be inserted, right? Just give me a second. If we go back to here, right? You see, the gene you're trying to insert, you should have flanking sequences that match the recognition site of the plasmid, right? So if your flanking sequence have the same recognition site as PST1, then your gene will be inserted here. Right? If it has the same recognition site as EcoR1, then the gene will be inserted here, right? So knowing where the restriction sites are will tell you where your gene will be inserted. What are AMP-R and TET-R? What does the plasmid have? Why does the plasmid have 2 of such sites? Okay, so these are, again, antibiotic resistant genes, right? Okay, antibiotic resistant genes. And why do you have two sites? Why not just have one resistant site? Why do you need tetracycline and ampicillin? Well, that has to do with the insertion site. Okay, if you insert the gene using PST1, then you actually interrupt the ampicillin gene, right? Then you will need the tetracycline for selection purpose. If you insert using BAMH, then you would disrupt the tetracycline resistant gene. Then you would be selecting using ampicillin. So by having more than one site, it gives you more versatility, right? As to where to insert the gene. Okay. All right. Now this is the confusing part. Let's take a look. Oh boy. All right, I'm gonna copy the map and we're gonna take a look at this. If you treat the plasmid with E-core R1, okay, so E-core R1 is right here, and then you run it on an agarose gel, how many bands do you expect to see? Oh, that's not the difficult question. Sorry, it's still quite easy. How many bands do you expect to see if you treat it with E-core R1? So if you treat it with E-core R1, it's going to cut right here. Then the plasmid will pop right open. You will see one band. Right? One band. Okay. Now here is the difficult one. If you insert the gene by treating your DNA and the plasmid with BAMH, okay, so BAMH is right here, BAMH, and transform it, feed it to E. coli, what are the expected results if you grow the transformed E. coli into the following culture? All right, so we're going to insert the gene with BAMH. It's going to go right here into the plasmid. Okay. Growth culture with no antibiotics. So if you put the E. coli on a culture with no antibiotics, do you expect them to grow or not grow? What do you think? Let's do a poll. I haven't poll all day long. Okay. Do you think it's gonna grow? If you think it's gonna grow, choose A. Grow. No grow. Is it gonna grow? Is it not gonna grow? What's it gonna be? All right. Now, if you put it in a culture with no antibiotics, no drugs, right? So just nutrients, they are of course gonna grow. Okay, so normal growth, because there's nothing there to kill them, right? So they will grow, regardless if they have the plasma or not. They will grow. Okay. Growth culture with tetracycline. Okay, will they grow in the presence of tetracycline? Well, because we are inserting the gene with BAMH, so the gene will go right here in the middle of the tetracycline resistant gene. In other words, it will interrupt this gene, right? That means if you grow them in a tetracycline culture, they will all die. So no growth. Because the gene that gives them resistance to tetracycline has been interrupted. What if you grow them in ampicillin? Are they going to be okay? They will be okay because the ampicillin resistant gene was not disrupted. So that will be normal growth. Does that make any sense? No? Okay. Can you ask a question? Okay. Let's backtrack a little bit. Do you guys all understand what AMPR and TETR means? Do you know what those means? No? Okay. You guys know antibiotics, right? Okay. Antibiotics kill bacteria. Okay. So that's first thing to know. Yeah, antibiotics will kill bacteria. So examples of antibiotics. Okay. So tetracycline is an antibiotic. Ampicillin is an antibiotic. Okay. Anything that ends with illin, right? Okay, or cyclin, right? Those are like antibiotics. Okay. Bacteria could protect themselves from antibiotics if they have resistance to it. Okay. So a bacteria is resistant to an antibiotic if the antibiotic cannot kill it. Is that okay? If bacteria is resistant to an antibiotic, it won't be killed. Won't be killed. Is that okay? All right. How does it become resistant? It can become resistant if it has the resistant gene. Okay, so these are resistant genes that will let them survive in the presence of the antibiotic. So if they have the AMP-R gene, they will be able to grow in ampicillin. If they have the TET-R gene, they will be able to grow in tetracycline. Okay, so AMP-R gene lets them grow in ampicillin. Okay, the TET-R gene lets them grow in tetracycline. So far, so good? Okay, let's backtrack a little bit more. What about these things? Do you guys understand what those are? PST1 equal R1, BAMH1, AVA1. Do you know what those are? Those are restriction enzyme sites, right? Yeah, those are enzymes, right? But more importantly, these marks the site of where the plasmid will be cut. Okay. And why do you want to cut the plasmid? You want to cut the plasmid so you can pop your gene in, right? Okay. Over here, right? Remember earlier we had a picture, right? Okay. You want to cut open the plasmid so you can insert this gene into it, right? Why do you want to insert the gene in there? If you insert the gene there and feed it back to the bacteria will now mass produce whatever protein this gene is coding for, right? So for example, if this gene codes for insulin, right? We can put it in a plasmid, feed the plasmid to the bacteria. Now the bacteria will keep on pumping out insulin, right? That we then can extract and, give it to people who need insulin. Does that make sense? So where are you going to insert the gene, right? Well, you can insert it in different places depending on your flanking sequence, right? Okay, so the flanking sequence is AATT. But you might not have an AATT in real life, right? Like you might have like, I don't know, like a CCGG instead, right? So this recognition site would not work if you have CCGG instead of AATT. Okay, so depending on what your flanking sequences are, you will have to choose different insertion site for the plasmid. So when you buy a plasmid online, okay, it comes with multiple insertion site. So if you cannot insert it here, then maybe you can insert it here instead, okay, or here or here. So it gives you 4 possible insertion site. It gives you more versatility, right, in terms of what kind of stuff you can insert. Does it make sense? Okay. So here is the question. Right? The question says, we will be inserting the gene by treating your DNA and the plasma with BAMH. All that means is, of all the possible insertion sites, location 1, 2, 3, 4, I'm going to insert it right here at the BAMH site. Okay. So what that means is everything else is going to be uninterrupted, but I'm going to put a gene right here. I'm going to put that gene I'm trying to clone to mass produce. I'm going to put it right here. Okay. Now, if I insert a gene in the middle of the tetracycline resistant gene, then the tetracycline resistant gene is not going to give the back. It's not going to work, right? Basically, because you basically destroy that gene. Does that make sense? Okay. If you insert the gene in the middle of the tetracycline gene, you're going to make it not work, right? You have to understand that before I proceed. Any question about that part? If you insert a gene in the middle of the tetracycline resistant gene, then that gene will, the tetracycline resistant gene will not work. Meaning the bacteria will no longer be able to resist tetracycline, right? That is why when you grow the thing in the presence of tetracycline, there will be no growth because the tetracycline will kill all of the bacteria. Okay? It is still okay to grow them in the presence of ampicillin because the ampicillin resistant gene was not disrupted, right? That's why they can still grow in the presence of ampicillin. Does that make more sense now? Yeah, so that basically is the answer, right? Now let me ask you a question, and anybody can answer, okay? What location would an insertion of a gene cause the bacteria to not grow in ampicillin? Where do I have to insert a gene so that they will die in the presence of ampicillin? Anybody? Yeah, if you insert it using the PST1 site, then it's gonna go in the middle of the ampicillin resistant gene, right? Now they will not grow in the presence of ampicillin because the gene was disrupted. Okay? Any other question? Okay, let's do another one. We are gonna insert using the equal R1 location. Okay, so equal R1 is right here. Okay, let's do a poll. Growth with no antibiotic will always grow. Okay, so growth, normal growth. Okay. What about in the presence of tetracycline? Okay, is it gonna grow in the presence of tetracycline? What do you guys think? Yes or no? Okay. Let's do a poll. Okay. If you insert using E core R1, will they grow in tetracycline?
Sorry, which one is yes, which one is no? A,
So
B.
A is yes, yeah. Yes,
Oh.
they will grow. No, they won't grow. C, confused. Let me know. Grow, not grow. Anyone else? Only 8 of you have answered. Where are the rest of you? Are you there? All right. So, tetracycline, which will normally kill the bacteria, right? But it's going to be okay if it has the tetracycline resistant gene. If you insert a gene in the equal R1 position, you will not be disrupting the tetracycline resistant gene. And so they will be able to grow, right? So growth, normal growth. Okay. What's the confusion? You know, the person who chose confused, what's the confusion? How can I address your concern? Okay, well, give me a question so I can help you understand. Okay, you can private message me or you can, you know, unmute, but I need to know what the confusion is before I can help you. What about growing them in ampicillin? Is it going to be okay? Yes, it's going to be okay. There will be growth because the ampicillin gene is not disrupted, right? If you insert it here, you will not disrupt it. Basically, this is the thing you have to know. If the location you're inserting your gene does not break up anything, right? Does not break up any of the resistant gene, then they will be able to grow in that antibiotic, right? All right, last question. Predict what will happen in each of the following. If you reverse the electrode, so the current runs from positive to negative, then what happens is all the sample will be lost. Right? All the sample will be lost. Okay, because instead of running down the gel, right? This is your gel. Instead of going this way, now they will be going this way. So you load the sample here. Okay, instead of going down, they will just all exit from the top, and the sample will be gone. Okay. B, run the gel for too long, the dye front is no longer visible. Okay, so what is the dye front? When you actually do this experiment, you cannot see the DNA at all. Okay, so again, it's hard to describe without actually doing this. But DNA is transparent, right? It's going to be in a solution. Okay, so you would add a little bit of blue dye, a little bit of blue dye. The blue dye will make the DNA sink into the well. And so, you know, you will see the blue dye, but you won't be able to see the individual DNA strand, okay? And so when you run the gel, you will see the blue dye, you know, going forward, and then everything else is going to be like transparent. You can't see the DNA, okay? But the dye front, this blue line, will give you an idea of where your DNA samples are. Generally speaking, your dye, which is very small, is going to be in front of your DNA sample. So all your DNA should not be past this line. So you will have your DNA like here or something, but you cannot physically see it, right? Okay. So the question is, if you run your gel for too long, right? Instead of running it for like half an hour, you run it for like, 2 hours. You forgot about it or something, right? Then what happens is the dye front exits the gel. That means, you will lose some of your DNA, right? So maybe the really short ones are gonna be gone, exit from the bottom. Okay. So some sample will be lost. Some samples will be lost. So how do you actually visualize the DNA, right? You will have to take the gel and you will have to soak it in something called ethidium bromide. Ethidium bromide is actually a carcinogenic substance. Okay, if you expose it, without protection, it might cause cancer. And the reason is because the ethidium bromide will insert themselves into the DNA backbone. Okay, and that's a good thing for what we're trying to do, because once they insert into the backbone of a DNA, they will glow in the presence of UV light. Okay, and that's how you can visualize these gels. They actually look like these things, right? These are like fluorescent that's given off by the ethidium bromide when you expose it to UV light. So if you forgot to add the ethidium bromide, then you cannot see DNA under UV light. Okay. and that's it. Okay, a lot of stuff to look through. And you know, the quiz is on Tuesday, but if you get confused about something, feel free to reach out to me. You know, I'm gonna try to sort it out with you. All right. I will post the information about the quiz. Yeah, Tuesday. Did I say Tuesday? Yeah, Tuesday. It is not Thursday. Yeah, Tuesday. Yep. So I'll post more information about the quiz later on. I will give you more time on this one because you know there are the pedigrees and stuff like that. So you probably have to you know work it out slowly. So you know I'll give you more time than normal. Well, Alex, the thing is going to be available from 6 a.m. to 11:59, right? You're going to be working the whole time for that. Anyway, maybe send me an email and then we'll work something out. Okay, it's yeah, I'm sure we can work something out. Okay, not to worry. Just send me an email, explain your situation, and I'll sort it out with you. Okay. And then like next week is the last one, right? Next week is the what is it? The tissue stuff, right? Okay. And then we'll maybe do a little bit of a test review as well. Okay. Yep, that's it. Thanks again. And I'll see you all next Thursday. Okay. Bye for now.